Good morning, everyone. Welcome back. Thank you for joining us. I'm Steve,
And I'm Scott.
and together we're Backyard Musings, the Science and Technology channel.
Yep, and we are talking about everybody's favorite topic in the hospital, and that is fungus.
And just on a side note, reunited, and it feels so good. I've got my laptop. It came back functioning perfectly. Beautiful. I'm just so happy I got it back, man.
No wonder you've been in a good mood.
A deadly drug-resistant hospital fungus may finally have a weakness, and scientists think they've found it. Researchers have identified a genetic process that could open the door to new treatments for a dangerous fungal infection that has repeatedly forced hospital intensive care units to close. The discovery offers fresh hope against a pathogen that has been difficult to control and even harder to treat. Would you like to know more?
Okay, here we go with some Latin, I believe. Candida auris poses a serious threat to people who are already critically ill, making hospitals particularly vulnerable to outbreaks. Although the fungus can exist harmlessly on the skin of many people, patients who rely on ventilators face a much higher risk of infection. That's not good. Once it takes hold, the disease kills around 45% of those infected and can withstand every major class of antifungal medication. This combination has made it extremely challenging to eliminate from hospital wards once it spreads. That sounds like COVID.
Yeah, or a staph infection. Yeah, sounds bad. On another side note, I have a horde priest, Pandaren female, and I named her Candida. How about that? All
Thanks for.
right. Candida auris was first identified in 2008, and scientists still do not know where it originally came from. Probably China. Since its discovery, outbreaks have been reported in more than 40 countries, including the UK. The fungus, also known as Candidozyma auris, is now recognized as a global health threat and appears on the World Health Organization, the WHO's critical priority fungal pathogens list. In the UK, case numbers have continued to climb over time.
Okay, so why have we not heard about this in the mainstream media?
I know, I don't know. I mean, it sounds like this is published from a UK scientist. That's what it sounds like.
Well, we share information with them. We're
Absolutely.
kind of
They're our
close.
cousins.
Yeah. Yeah, this is crazy. For the first time, a team at the University of Exeter has closely examined how Candida auris activates its genes during infection using an innovative experimental model based on fish larvae.
Oh,
Research was published in the Nature Portfolio Journal Communications Biology and received support from Wellcome, the Medical Research Council, MRC, and the National Center for Replacement, Reduction, and Refinement, NC3R. Interesting.
The results suggest a promising path toward identifying a biological target that could be used to develop new antifungal drugs or adapt existing ones, provided the same genetic activity occurs during infection in humans.
The work was co-led by NIHR clinical lecturer Hugh Gifford from the University of Exeter's MRC Center for Medical Mycology. He said, quote, "Since it emerged, Candida auris has wreaked havoc." Again, why aren't we hearing about this? Where it takes hold in hospital intensive care units. It can be deadly for vulnerable patients, and health trusts have spent millions on the difficult job of eradication. We think our research may have revealed an Achilles heel in this lethal pathogen during active infection, and we currently need more research. Of course they do. Well, more the merrier. Get on it to explore.
Give me your money.
Well, yeah, get on. Yeah, this is terrible. Whether we can find drugs that target and exploit this weakness, unquote.
Yeah, this sounds nasty.
Yeah.
I am really surprised we haven't heard of it. But you heard it first here on Backyard Musings, Science and Technology. One long-standing challenge in studying Candida auris is its ability to survive high temperatures. Combined with its unusually strong tolerance to salt, this has led some scientists to speculate that it may have originated in tropical oceans or marine animals, such as found in China. I threw that in. I just threw it in. These traits have also made it harder to study using traditional laboratory models.
To overcome this, the Exeter researchers developed a new system using Arabian killifish, whose eggs are able to survive at human body temperature. This allowed the team to observe the infection process in a living host under realistic conditions. So, killifish, huh? Never heard of that. Arabian killifish. Okay.
During the study, the researchers observed that Candida auris can shift into elongated fungal structures called filaments, which may help it search for nutrients inside the host. They also tracked which genes were turned on and off during infection, highlighting potential weaknesses. Several of the activated genes are involved in producing nutrient pumps that capture iron scavenging molecules and pull iron into fungal cells. Iron is essential for survival, and this dependence could represent a critical vulnerability. Well, we got iron in our blood.
Yeah. Co-senior author Dr. Rice Farrer from the University of Exeter's MRC Center for Medical Mycology said, "Until now, we've had no idea what genes are active during infection of a living host. We now need to find out if this also occurs during human infection. The fact that we found genes are activated to scavenge iron gives clues to where Candida auris may originate, such as an iron-poor environment in the sea. It also gives us a potential target for new and already existing drugs."
Dr. Gifford, who also works as a resident physician in intensive care and respiratory medicine at the Royal Devon and Exeter Hospital, emphasized the potential medical impact of the findings. He said, quote, "While there are a number of research steps to go through yet, give me your money, our finding could be an exciting prospect for future treatment. We have drugs that target iron scavenging activities. We now need to explore whether they could be repurposed to stop Candida auris from killing humans and closing down hospital intensive care units."
That would be devastating.
Yeah.
The Arabian killifish larva model was developed with the support from an NC3R's project grant as an alternative to using mouse and zebrafish models, which are commonly employed to study how pathogens interact with their hosts. Dr. Katie Bates, NC3R's head of research funding, okay, so she's going to ask for money, said that this new publication demonstrates the utility of the replacement model to study Candida auris infection and enable unprecedented insights into cellular and molecular events in live infected hosts. This is a brilliant example of how innovative alternative approaches can overcome key limitations of traditional animal studies. Okay. Yeah, this is bad stuff here. We need to
There you have it. You heard it first from Backyard Musings.
Yeah, we need to really get on this and take care of it. I mean,
yeah, we're going to keep an eye on this and see where it goes, and hopefully we'll get more news about it.
Yeah, absolutely. Okay.
All right, folks. Thanks for joining us. We'll catch you this afternoon.
Take care. We'll see you this afternoon.